Okay. So thank you very much for the invitation, Danny. Our lab is a new lab, so we've just entered it 2 weeks ago. And that gives me an excuse to talk a lot about things that have just started and also about things that are in the planning. I want to tell you about two things. One is the type of questions we're interested in our lab, and two is how we think microfluidic devices can be used effectively to answer some of these questions. So in our lab, we're interested in how cells make decisions. And specifically, we're looking at cases where groups of cells, similar cells to begin with, meet some external condition or some signal from the environment. And upon this signal, some of them decide to change their state in a significant way. And we know many examples of such processes of change from fungi, bacteria, up to mammalian cells in all kinds of systems. But if we look at the snapshots of these change processes, the one thing we see is that different cells respond differently, even if the external conditions are exactly the same. So in this differentiation assay, some cells will differentiate while others not. Here, under certain burn conditions, some cells will die when others not. Some grow filamentous while others stay round. And some turn into a process which I'll describe in more detail and develop those balls, and the others stay growing normally. So we're interested to ask what kind of decisions or responses cells can make. What are the mechanisms and factors leading to different responses? And in a more mechanistic or information perspective, how do the cell systems or networks process information from the environment to reach these kinds of decisions? We work in two different biological systems. One of them is yeast in a process called meiosis, which I'll describe in a second. And the second is in mammalian cells in processes of reprogramming and differentiation, which we've already heard a lot about this morning. So I'll start with the first system, in which this question is a bit simpler, because in the specific system that I'll describe to you, all the cells go through the same decision or through the same response. That is, they all eventually turn into meiosis and sporulation. The only difference between them will be the time of response or the time of decision. So without going into too many details, a yeast cell, as long as it sees food, it rotates happily in the cell cycle. But when it senses starvation, it can go into this emergency program called meiosis and sporulation, which can save it from bad times. There is a point which can be naturally defined as a point of decision, and that is the point in which it becomes detached from the signals, and even if you bring food back to the environment, it will keep on in the same process through the bitter end. And so we'll define this as the decision point, and we'll try to ask when do different cells commit, and what are the things that control this time. So I'll describe our approach, the experimental approach, and then you'll see how microfluidics fits in very naturally in it. We use fluorescent markers to tag genes which are related to the process, specifically turned on very early in the meiotic process. And then after growing the cells happily, we give them the signal, and we do this in a kind of a flow cell, which allows us to control the conditions and give all the cells in the experiment exactly the same conditions and uniformly over time. And this is mounted on a microscope with an automatic stage, so we can actually take several dozen movies, which look something like this. At the beginning, so this is the fluorescent channel. So at start, we don't see anything because the process hasn't started yet. But as the cells start to sense the starvation, they turn on their signals one after the other, and then at some point you'll see these two splits that look like fireworks, which are the two meiotic divisions, and we can associate the decision time with one of them. So this is one, if you multiply this by 50 of those movies are an output of one typical experiment. And of course, we need some sort of a tool, computational tool to deal with that. So with some image analysis code we wrote, we can turn this movie into quantitative tracks in time from individual cells. And a bit more than that, we can actually even detect those splits and actually the decision events, and we can annotate them automatically for all of the cells. Okay? And if we're able to do that, we can start to ask questions about what is the distribution of decision times. The answer here is very broad. So compared to the cell cycle, which only takes an hour and a half, cells can commit from anywhere between 7 to more than 24 hours after you give them the signal. So this is a puzzle why they need to do that. We can increase a bit the resolution and enter another measurement point, which is the time the early meiosis genes kick in, and we can further split this interval and ask how long they wait and how long they actually start and progress in the process. And the surprising answer is they wait most of the time. I'll show just one more result in showing that we can connect between molecular levels and phenotypes. So if we mark one particular gene, which is a master regulator, and we look at its molecular level, we can try to ask how does this molecular level, which is a bit different between different cells, in this case it indicates the promoter activity or the production rate of this protein, how predictive is this about the entry time or the decision time? So we can use two fluorescent markers with different colors for this purpose, and we can look at correlation and find a very strong connection. So molecular noise or molecular level affects the entry time, the decision time. Okay? So I won't go more into the details here, but using, again, some statistical tools, and the fact that we have measurements from thousands of cells, which gives us the power to do statistics, we can draw more of these causal connections and put numbers on them. So how much of the variance in this parameter is governed by, for example, cell size and so on. We can rule out some of the factors, for example, the cell cycle phase or the nutrient signal integration are not significant factors in determining the decision time. We can do some more elaborate tests, which I won't go into. And this combination of live imaging and statistical tools is the basis for a platform. So since this is a nanos conference and I was foolish enough to put the word micro in the title of my talk, I have to show that even in the large scale and high throughput manner, we can also observe submicron structures. In this case, the yellow dots are what's called spindle pole bodies, and are, I think they're less than 100 micron in diameter. Nanometer, sorry. And we can actually, even in this poor resolution, we can do some statistics with them. Okay. So moving on to new questions. So far, all I've showed you is pretty descriptive or phenomenological, but behind all this, of course, there are signaling pathways and transcriptional pathways that mediate between the environment or the signals in the environment and the actual processes that happen in the cell. So there are three projects running now in the lab, all using microfluidics based on these three levels: signal processing, master regulator, and transcriptional program. So I'll briefly describe each of them. So far, in the experiments I showed you, the cells were subjected to a continuous signal, but life in nature sometimes is more complex. Sometimes there are fluctuations in the signal. The temperature and humidity can change between day and night, and so on. And it might be interesting to ask how do cells respond to fluctuations in the signal? How does this affect the distribution in their population of behaviors? The flip side of the coin, which might speak to the engineers here, is fluctuation is a powerful tool to study the dynamic properties of systems. So you can actually use it to study specific components in circuits and how they behave dynamically. So we can ask a whole slew of questions regarding signal perturbations. Can cells filter out noise from the environment? Can they integrate over time the signal? And so on. And if we had some way or some tool that will allow us to probe different heights of a pulse, of a spike or a perturbation in the signal, and different durations, that would allow us to sort of explore this phase space of responses of cells to perturbations in the signal. And the way we do this is by replacing the macrofluidic cell with a microfluidic chamber. So this is a basic microfluidic chamber with two inputs and one outputs. In all our chambers, there is one big channel, which is basically the imaging chamber. This is where we immobilize the cells, and this is where we image them, like in the movies I showed you before. And for example, here, if we have two inputs, in one of them there is the signal, and we open and close them alternatively, we can generate a spike in the signal. The good thing about microfluidics is we can very quickly generate new designs, and within a day or two have the chips ready in the lab. So for example, we can look at different conditions in parallel. We can do different spikes and measure them in the same experiment. And we can use these more elaborate designs invented in the Whitesides group at Harvard that actually use the property of, they can actually generate a gradient along the width of the channel. And this will actually give us a tool to probe different concentrations. So if we take this last design and we turn on and then off the gradient, we can generate a spike with different heights. If we take the simple wide channel and we use the property of laminar flow, which generates a sharp boundary between the two inputs, we can play with the pressures of the two inputs gradually and therefore change the boundary, and this is a real movie, and then turn off the whole thing. And if you think about it, cells that sit here show a longer spike than those that sit here, and so on. So we can actually have a range of durations of spikes in the same microfluidic device in cells sitting side by side. The take-home message is microfluidics allow us flexible and dynamic control over the inputs, and we can use it to ask different questions. For example, how is a spike of glucose affecting the decision time? And this is just one sample result. And then we can ask, how is this perturbation, how is this information propagated through the signaling and transcriptional pathways? And for this, we can actually use different fluorescent markers, give a spike of glucose, for example, and see how it propagates in those different levels. Okay? Right now, we're in the process of implementing this system to study crosstalk and signal merging between two very important and central signaling pathways that are conserved all the way from yeast to human, the PKA and the TOR pathway. There are very interesting questions to ask there. So to touch quickly on the second level, that's the level of the transcription, the master regulator. This is in all those cases, this is a very central protein, and often it has many levels of regulations. In this case of meiosis, there are multiple positive and negative feedback circuits working on it. And with tools, we can use different genetic strains, deleting one or two copies of the different components, And using this way combined with the live imaging, we can actually probe the dynamics of these different feedback levels. So for example, we can see when comparing the no feedback case in blue to the yes feedback case in red, that positive feedback happens early on, then they two sort of cancel out, and then at around 20 hours, negative feedback kicks in. Okay? And our idea is to use the microfluidic devices to spike signals in different epochs, the early, late, and middle, and see how the system responds in different regimes of the feedback circuits. And lastly, for this system, the transcriptional program, if you think about it, a master regulator in this case activates dozens or hundreds of genes. These genes, the proteins encoded from them, have to function in different steps of the process. For example, generating double-strand breaks in the DNA, doing recombination. These processes have to happen in a certain temporal series. The question is whether a single regulator can exert enough control over its targets to maintain a certain time difference between them, or a certain stoichiometry between them. For example, if they are complex members. So we can use multiple colors in the same cells, for example, tagging two targets or a target and a regulator to measure these things in single cells over time. Okay? So for a few last minutes, I want to switch to the different system, which is the mammalian cell system. And we heard a lot from Daphne Benayahu this morning about differentiation in stem cells. We currently have projects both on reprogramming and differentiation. And I'll try to briefly explain what these processes are in very general terms. So in our body, or in the female body, a single fertilized egg develops into a full embryo, and at some point, early point, there is a group of cells called embryonic stem cells, which are in a single, are thought to be in a single defined state, and through development, they give rise to all the cells in our body. So skin cells, heart cells, cardiac cells. They all go through a series of differentiation steps. We can try to emulate this process in vitro in the dish, and some of these efforts Daphne described this morning, toward a specific fate, for example, toward the heart cell. But as we heard, we have problems of efficiency. We don't know often what are the mechanisms, what are the compounds we need to do to induce this kind of change. And there's an inherent difference between standard two-dimensional culture conditions and what's actually happening in an embryo, which essentially is a three-dimensional environment. A recent breakthrough of 3 years ago is that we can actually take an adult cell, a skin cell, and through molecular manipulation, trick it into, after waiting a long time, about 2 weeks, and growing it in a dish, trick some of its progeny to going into a state which is very, very similar to an embryonic stem cell state. So this is called reprogramming. It has immense importance, as far as we know, for regenerative disease and for basic research. Again, here we have similar problems. The efficiency is very low. And here, as opposed to development, we know almost nothing about this process. We don't know what's actually going on there. So we started by looking at this process in a collaboration with a lab in Harvard that's working on reprogramming, using similar approaches. So this is a whole reprogramming process, about 2 weeks worth of imaging. And this is actually stitching of several positions. So this is about a quarter of a well like this. And what I want to show you here is the tool of a fluorescent tagging of cells used for lineage tracing. So we start with some mixture of cells with a green fluorescent or yellow fluorescent or red fluorescent protein, and each one of them gives rise to some progeny. Okay? So for example, these guys successfully reprogram and they become what we want, but the guys stemming from this yellow progenitor growing to this colony turn into something closer to cancerous cells. Namely, they proliferate a lot, but they don't become pluripotent like stem cells. If we use immunofluorescence, immunofluorescent tagging at the end of the process, we can, with some certainty determine which are the successful colonies in this process, and then backtrack to analyze these colonies. And again, we use image analysis and some computation, and we can sort of define different behaviors. The geniuses, the ones that do the correct work, the ones that just fast divide, like cancer cells, the ones that slow divide. And these are the columns here, and each row is a certain thing that we measure. For example, growth rates, log number of cells, the cell area over time, the eccentricity of the cell, how round it is or how elongated it is. And we can, using these characterizations, we can actually find that the decision whether to go this or this or this way happens very, very early. And it might even be predetermined as far as we know. And we're actually now looking for molecular markers. Daphne discussed the difficulty of this morning to help us distinguish the very early phases of decision, how to go to this route or this route. Remember, I talked about the problem of 2-dimension versus 3-dimension. About 2 years ago, a group used a microfluidic device, a group from Berkeley University, to study clusters of cancer cells. And their purpose was to generate these microtumors and to study how drugs penetrate into those microtumors and how they affect them. And the idea is to use the same microfluidic channel as I showed you before, but to have these U-traps. And by controlling the size and geometry of the U-traps, you can control how many cells, what's the size of the cluster, and what's its geometry. From my perspective, this also gives us an option to look at a large number, several hundreds or thousands of instances. In this case, every instance is a cluster of cells, and we can follow differentiation dynamics in them. So the plan is to use, again, fluorescent lineage markers. If we find good cell state markers, to use them, put the ES cells in this kind of device, follow them over time and suggest rules for differentiation, and then maybe we can use some interventions like small molecule or siRNA to shut down or activate some signaling pathways and see how it affects the result. Just to end with a more future idea of using microfluidics, it turns out you can actually have a peristaltic pump on a microfluidic chip. In the Quake Lab, which are the masters of doing things at large scale in microfluidics, actually have chips with hundreds of these kinds of pumps. They even do chemical reactions in them. All these things are switches, and if you turn them on in a certain sequence, you create a peristaltic pump. But my idea is to use these to follow individual cells in a blood-like flow. And you can control the parameters of the flow by controlling the speed and the thickness of these channels, and to look at processes which actually happen in flow, for example, infection by malaria parasite or viruses. And this is more distant future. Just to summarize, we look at developmental decisions in cell populations, and through the use of microfluidics, we can both control the dynamics, the inputs flexibly. We can generate controlled growth geometries in cell clusters, and we can emulate natural conditions, for example, flow environments. So just to end with, this wasn't intended to be subliminal, so to thank my collaborators in Harvard, the stem cell reprogramming people, Alex and Zach, my former PI at Harvard, where all the yeast work started, and my current lab members, and a special plea for the southern side. If you think about your next step and you like live imaging and a combination of microfluidics, modeling, image analysis, or any subset of these, please contact me.
You got money?
Hmm?
You got money?
Yeah. Thank you.
Okay, 2-3 questions. Oli? Do cells know to count signals? Some decisions are made sometimes after several signals already.
So it's a very good question. I haven't done these experiments yet. There have been works looking at the SOS system in bacteria and also in the P53MDM2 system, a central protein for anti-cancer protein, showing that these systems actually generate series of pulses, series of responses. So, but these are actually cases where the genetic circuit itself is generating a discrete number of pulses. So actually, I don't know of experiments that try to do that. It's a good question. Yeah.
Okay, thank you very much.
Thank you.